is Gulnara Tagirdzhanova. I think I've got that a little bit wrong, I'm sorry. She has a master's degree from the St. Petersburg University in Russia, where she worked on lichen conservation biology. That is the end of the last talk. Sorry, that was my phone saying the timing was up. Sorry, Gulnara. She has a PhD. She's a PhD candidate now at the University of Alberta in Canada. She's working in Toby Scribley's lab on the metagenomics of lichen symbiosis. So I'm looking forward to this one too. Thank you, Gulnara.
Hello, my name is Gulnara Tagirdzhanova. I am a PhD candidate at the University of Alberta. And my talk today is about genomes of lichen-associated yeasts and what we can predict from them. Until recently, we thought that most lichens consist of two partners, a fungus and an alga. In this dual model of lichen symbiosis, it is believed that the alga supplies the fungus with carbohydrates, and the fungus, in turn, creates a symbiotic structure to host the algal cells. Now we know that in some lichens there are some additional secondary symbionts in the form of yeast or bacteria. They are present in lower cellular abundance compared to the two dominant partners, but they are present consistently. And the question is, how do they fit into the lichen symbiosis? What is the relationship between them and the dominant partners? And do they contribute anything to the system? There has been several hypotheses made that they are mutualistic symbionts, that they are parasites, but until recently there was very little evidence one way or the other. One of the reasons for that is that these yeasts are very hard to study. We haven't got them into pure cultures, and therefore we cannot study them experimentally. A usual alternative would be metagenomics. Metagenomics gives us a set of methods to study microbial communities by sequencing intact samples. So this way we don't have to separate individual lineages, we can get genomes of multiple organisms at once. However, the secondary symbionts in the lichens are present in such low abundance that it's really hard to get enough data from them. And in addition to that, lichen symbiosis is so complex that the resulting metagenomic data is really hard to deal with. As a model system for our study, we chose Alectoria sarmentosa. It is a macrolichen that consists of two dominant partners, a Lechanoromycete from the family Bromeliaceae and the green alga. Also, one secondary fungal symbiont has been reported from Alectoria, the Cyphobasidium yeast. Here is our protocol summed up. We generated two metagenomes. One of them from an intact fragment of a lichen, we called it bulk lichen metagenome, and the other from the sample we called cortex slurry. So how and why we made this cortex slurry? From the previous research, we knew that the yeasts live in lichen cortex. They are embedded in the extracellular matrix there. So we used hot water to dissolve this matrix. Then we concentrated the solution and used the resulting slurry to make a metagenome. Then we analyzed the data, and I won't go into details about the bioinformatics now, but I'd be happy to answer any questions. On these plots, each colored blob represents a genome. So in the case of bulk lichen metagenome, we got two genomes, the Lechanoromycete and the alga. There also were little traces of yeast genomes mixed in, but really that was not enough to study them. And in contrast, in the case of the cortex slurry metagenome, we got genomes of multiple secondary symbionts here and here. So this confirms that the cortex slurry helped us to achieve our goal of increasing the abundance of the secondary symbionts, and therefore helped us to acquire high-quality genomes from them. In total, we extracted 3 fungal genomes from the cortex slurry metagenome, the Lechanoromycete fungus and Cyphobasidium, and this is what we expected when we started the study, but then there was another genome that we expected less, and it was from Tremella. Tremella as a group includes many lichen-associated fungi, and until recently, all of them were thought to be parasites. From a recent paper, we know that Tremella is consistently present in Litharia lichens in the yeast form. And now we also found it in Alectoria. So this is our final dataset. 3 fungal genomes extracted from a metagenome. Each of them had high quality comparable to what you can expect from a genome sequence from culture. We annotated each of the genomes, meaning that we predicted genes and assigned those genes with putative functions. Okay, so now we can get to the part where we look into these genomes and try to understand what can they tell us about the biology of the fungi. We looked into many aspects, and now I wanted to share with you just 3 highlights. And the first is secondary metabolism. We looked into genes related to the secondary metabolites biosynthesis. And this graph shows the number of these genes in different genomes. And you can see that the Lecanoromycete from Alectoria sarmentosa had many, many more of those genes compared to both Cyphobasidium and Tremella. So we can hypothesize that the Lecanoromycete is most responsible for the secondary metabolite production in the Alectoria lichens. The second class of proteins we looked into were carbohydrate active enzymes, or Kzymes. Those enzymes are involved in producing, modifying, and breaking down polysaccharides. And we became interested in polysaccharides because they are the main component of the matrix of lichen cortex. So effectively, it is them who glue the cells of lichen symbionts together to achieve the lichen form. And also the yeasts are embedded in those polysaccharides. And there is a lot to talk about Kzymes, and I could make an entire talk just about them. But for now, I wanted to focus on one example. Both the yeasts, but not the Lecanoromycete, have genes responsible for synthesis of glucuronoxylomannan, or GXM. It is a polysaccharide that is known from other non-lichen tremellas, and it is extremely hygroscopic. Tremella fuciformis, for example, here, owes its common name, the jelly fungus, to this polysaccharide, and the moisture-retaining properties of GXM are even used in cosmetics. So here you can see a skin lotion made from tremella, and all thanks to GXM. So now we can hypothesize that maybe the yeasts in lichens do produce GXM, and that helps lichen to retain water. Finally, we cannot answer our burning question. Are the yeasts mutualistic or parasitic? It turns out that genomic profiles are not really enough to answer this question. However, there was some evidence I wanted to mention. First, tremella has a profile of a nitrogen oxytroph. It means that it cannot assimilate inorganic nitrogen. It has to get organic nitrogen from some other living organism. But we cannot tell if it gets it from the Lecanoromycete, from the alga, or from somewhere else. Second, all 3 fungi have putative polyol transporters. And it is important because polyols, also known as sugar alcohols, are the carbohydrates that the alga delivers to the Lecanoromycete. And third, Cyphobasidium produces enzymes that target and break down plant polysaccharides. So taken together, this evidence led us to hypothesize that the yeast might rely on alga for sugars, and this can be viewed as evidence against the hypothesis that the yeasts are parasitic on the Lecanoromycete. Okay, to sum up, we used a combination of a wet lab technique we called cortex slurry and bioinformatic analysis to obtain the first 2 genomes of lichen-associated yeasts. Based on these genomes, we generated a testable hypothesis about the metabolic potential of the yeasts and about the trophic strategy. And this is the first ever evidence on the role these yeasts play in lichen symbiosis. And with that, I wanted to thank all the collaborators and thank you for your attention.
That is fascinating. There's lots of, there's so many questions a person could ask about that work. And there are so many aspects that you have to be an expert in nowadays, aren't there? So thank you for a really clear talk. I'm going to leave it to Clara and you to have some time for questions.
Hello.
All right. Hello. Yeah, fantastic. We are waiting for some questions to come in. I think maybe everyone is kind of feeling like me, just blown away by the exciting information you just shared. But I guess one question is, are you hoping to do this with other species? Now that you've looked at this one really in depth, are you going to look at other lichens?
Ultimately, yes. Yes, that will be the goal, to try to cover the diversity and different lichen systems, and hopefully maybe even, different origins of lichenization, so to say, look at basidiomycete lichens and all of that. Yeah, so we are not quite there yet, but hopefully one day.
Yeah. Okay, so we have a question from Julia Gerasimova. What approach did you use to separate and identify the fungal genomes?
Yeah, thanks for the question. Hi, Julia. Yeah, for that actually, let me see if I can do that. Like I have actually prepared a slide to share, and I have like a zillion of tabs open. So which one is the right one? Okay, so there are with different binning approaches. There are two main ways of doing it: database binning and database independent binning. And we chose database independent approach just because we suspected that for, say, Cyphobasidium, there hasn't really been any genome sequenced yet, so comparing your genome to like a database of all existing fungal genomes can just not really do the trick if the databases don't include species that your metagenomes include. So instead, we used like basically two statistics. One is so-called oligonucleotide distribution of sequencing content. It's basically, the idea is that different genomes of different organisms tend to have different structure in terms of, like how many G's or C's or like different small nucleotide words they have in the genomes, what are the distribution statistics of those genomes. And also we used coverage, and that just to separate like higher abundance organisms from lower abundance organisms. Sorry, that was like a long-ish answer.
No, it's okay. That's really interesting. So from Nathan, we have a question. Have you looked at chitinases in the yeasts and whether that might help answer the question of if they're acting in a parasitic sort of way?
Yes, no, that's a great question. We did look into that. We looked into like all enzymes that potentially might be targeting fungal polysaccharides, but the problem is that all fungi produce some chitinases that they use to remodel their own cell walls, and it's really hard to say like what's the purpose of this exact chitinase for this exact fungus. Yeah, we did look into that though.
Awesome. A question from Anne Evancout. Did you would you expect other Tremella species in other individuals of the same microbiont?
Oh, okay. I actually have a slide prepared for that too. Yeah, we are working on, we have a paper under revision, and there is like all the illustrations and images that I made for it, that I cannot include all of them in the talk, but I really want to share. So here we did some PCR screening just to test for that, just to make sure that the, like, say, Tremella and Cyphobasidiums are actually present consistent in Alectorias and not just some hidden miss. So we collected 33 other Alectorias and paired them with other specimens of like other lichen species that grow close to Alectoria, and also just tree bark, just to make sure that what we are seeing is not like random contaminants from like environment and all of that. And here you can see it's a little bit busy, the plot. And the top panel shows Alectoria, and you can see that, yes, Cyphobasidium and Tremella are consistently present. And I also have a bunch of trees based on marker genes to see, and yeah, mostly they, like I saw that, Cyphobasidium and Tremella from Alectoria grouped together with lineages from Bryoria and other Alectorias. So it's like, yeah.
Awesome. There's a question from Andreas Beck. Could you already look into transcription patterns of these lichen-associated yeasts?
I think so, yeah. It's a little bit hard because, again, of this very unequal abundance problem. But in principle, yeah, that would be like a potential next step to look on these genes and whether they actually expressed or not.
Great. Thank you, Gulnara. There are more questions coming in, so I invite you to look through that question panel. Wonderful. Thanks for your talk.